Good morning, everyone, and thank you for being here today. I am pleased to introduce my presentation titled Statistical Optimization of In-Process Sampling During OSD Manufacturing. In this session, we will explore how statistical methods can improve sampling processes in the manufacturing of oral solid dosage forms. We'll cover key techniques, benefits, and practical insights to enhance quality and efficiency. Let's dive in and see how these approaches can make a real difference in our manufacturing processes. In the world of oral solid dosage manufacturing, or OSD, in-process testing, or IPQC, plays a crucial role in ensuring product quality. During production, whether it's blending, compression, or coating, we need to take samples at various stages to monitor the process. However, this sampling comes with some challenges. For example, taking too many samples can lead to material loss, which isn't ideal, especially when dealing with expensive or limited materials. It also means more time and higher costs for analysis, which can impact overall efficiency. Plus, excessive sampling can increase batch wastage, affecting our production yield. Now, regulatory requirements add another layer of complexity. They mandate that sampling must be scientifically justified and truly representative of the entire batch. This ensures that our testing results are reliable and that we're compliant with regulations. Our goal here is clear. We want to optimize our IPQC sampling strategy. We need to find that balance where we're maintaining product quality and meeting regulatory standards, but without unnecessary sampling that could lead to waste or increased costs. Ultimately, it's about making our process smarter, more efficient, and still fully compliant. Regulators require a careful and thoughtful approach to sampling, focusing on risk-based sampling strategies. This means that sampling should be prioritized based on the potential risks involved, ensuring that we target the most critical areas. It's also essential to collect representative samples, which truly reflect the batch or process we're evaluating. This helps ensure our results are accurate and meaningful. Proper documentation of sampling plans is another crucial point. Clear records not only support compliance, but also provide transparency and traceability of our sampling activities. Now, looking at the regulatory framework, there are several key regulations and guidelines that govern in-process sampling. For example, 21 CFR 211.11 emphasizes the importance of sampling and testing in-process materials to maintain quality. Similarly, 21 CFR 211.16 advocates for scientifically sound sampling plans, underscoring the need for rational and justified methods. On the international side, EU GMP Volume 4 and WHO GMP set standards that align with these principles, promoting consistent quality across regions. Additionally, ICH guidelines like Q8, which covers pharmaceutical development, Q9 on quality risk management, and Q10 outlining the pharmaceutical quality system, all play pivotal roles. They collectively emphasize the importance of scientifically justified, risk-based, and well-documented sampling practices to ensure product quality and compliance. Connecting all these points, it's clear that a robust sampling strategy, aligned with these regulations and guidelines, is fundamental to maintaining quality throughout our manufacturing process. ICH Q9 recommends making risk-based decisions to help us optimize our control strategies. This means we focus our efforts where they matter most, based on the level of risk involved. When we talk about risk-based sampling, we're essentially targeting testing on those critical process parameters, or CPPs, that directly impact the critical quality attributes, or CQAs. So, instead of testing everything equally, we prioritize the areas that could have the biggest effect on quality. To do this effectively, we use tools like FMEA, which helps us identify potential failure modes and their effects. Hazard analysis allows us to spot potential risks early on, and process capability studies give us a clear picture of how well our process performs in relation to those critical parameters. All of these tools work together to ensure our sampling strategy is aligned with the actual risks, helping us maintain quality efficiently and proactively. This approach not only supports compliance, but also enhances overall process understanding and control. Let's talk about the statistical optimization approach. Essentially, it's about using statistical sampling to get reliable results while keeping the sample size as small as possible. This way, we can save time and resources without sacrificing accuracy. Some common statistical tools we rely on include ANSI ASQ Z14 and ISO 2859 sampling plans. These are standardized methods that help us decide how many samples we need to test. We also look at process capability indices like CP and CPK, which tell us how well a process meets specifications. And confidence interval sampling is another technique that helps us estimate the true process performance within a certain level of certainty. The key point here is that these standards and tools help us determine the minimum number of samples required to maintain statistical confidence. In other words, they guide us to make smarter sampling decisions, ensuring our results are reliable without unnecessary testing. This approach ties into the overall theme of quality assurance, using data-driven methods to optimize our processes efficiently. Now, let's take a closer look at the typical stages involved in manufacturing different pharmaceutical forms. For each stage, we perform IPQC sampling to ensure quality control throughout the process. Starting with capsules, the main steps include capsule filling, polishing, and packing. Each of these steps is critical to maintaining product integrity and ensuring that the capsules are correctly filled, free of defects, and properly packaged for distribution. Moving on to dry syrup, the manufacturing process involves blending the ingredients thoroughly, then filling the blended mixture into containers, and finally packaging the finished product. This process emphasizes maintaining uniformity and preventing contamination at every stage. When we consider tablet manufacturing, the process begins with dispensing the raw materials, followed by granulation, where powders are transformed into granules for better flow and compression. Then comes drying the granules, blending them uniformly, and compressing the mixture into tablets. Coating may follow to improve stability, taste, or appearance, and finally, the tablets are packed for shipment. For capsule manufacturing, the key step after blending is filling the capsules with the prepared powder or granules, ensuring each capsule contains the correct dosage. Throughout all these stages, IPQC sampling plays a vital role in monitoring quality, catching any deviations early, and maintaining high standards across every batch. This systematic approach helps us deliver safe and effective pharmaceutical products to patients. Let's take a closer look at stage-wise sampling versus wastage, which is what we often see in typical industry practice. In many cases, companies tend to use stage-wise sampling as a way to manage quality control across different phases of production. This method involves checking samples at various stages rather than waiting until the end. The idea is to catch issues early and reduce the risk of large-scale wastage later on. However, despite its advantages, this approach can sometimes lead to increased wastage, especially if the sampling isn't representative or if mistakes are caught too late in the process. It's a balancing act. While stage-wise sampling helps identify problems earlier, it can also result in unnecessary rework or discarded materials if not managed carefully. Connecting this to our overall theme, it's clear that the goal is to optimize quality control without incurring excessive wastage. This slide highlights the importance of understanding industry practices so we can develop more efficient, less wasteful methods. Ultimately, by improving our sampling strategies, we can better control quality while minimizing wastage, driving both efficiency and sustainability in our operations. Let's take a look at some of the key benefits of this optimized sampling approach. First, we're seeing a 40 to 50% reduction in the sampling quantity. That means we're using much less material overall, which can lead to significant cost savings and more sustainable processes. At the same time, this approach helps lower material wastage, so we're not only saving resources, but also reducing waste management concerns. The core of this strategy is an optimized sampling plan, which is developed through process validation and supported by solid statistical justification. By applying these scientific methods, we ensure that our sampling is both efficient and reliable, giving us confidence in our quality control while minimizing unnecessary sampling efforts. Overall, this approach aligns perfectly with our goal of creating smarter, more sustainable, and cost-effective processes without compromising on quality. Let's take a closer look at the reduction in sampling wastage. We've achieved a 66% decrease, which is quite significant. Now, to illustrate this, we'll walk through an example of tablet compression sampling optimization. Imagine we're working with a batch size of 500,000 tablets. In the traditional approach, we sampled 30 tablets every 30 minutes over a 10-hour production period. That adds up to 20 sampling intervals, so in total, we take 600 tablets for testing. Now, with the optimized approach, we're sampling fewer tablets, just 20 tablets, but doing so less frequently, every 60 minutes. Because we have fewer sampling points, only 10 intervals, the total number of tablets sampled drops to just 200. This example clearly shows how adjusting our sampling strategy can dramatically reduce waste without compromising quality. It ties back to our overall goal of making our processes more efficient and sustainable. Let's talk about sampling locations first. It's important that they represent different depths and positions within the blender. This way, we get a more accurate picture of the entire batch, rather than just one spot. Moving on to reduction, we've achieved a 66% decrease in blend sampling wastage. That means we're using significantly fewer samples to get the same level of quality assurance, which saves time and resources. Now, let's look at the example of blend uniformity optimization. Traditionally, you might take around 30 samples, each weighing about 5 grams. That adds up to a total of 150 grams of blend used just for sampling. With our optimized statistical method, we're reducing that number dramatically, taking only 10 stratified samples. The total blend used drops to just 50 grams. This approach not only maintains the accuracy of our quality checks, but also makes the process much more efficient and cost-effective. It's a great example of how applying proper sampling strategies can improve overall process efficiency without compromising quality. On this slide, I want to highlight a significant achievement we've made in reducing waste. We've managed to cut sample wastage by 60%, which is a substantial improvement. This reduction is backed by our process validation data, ensuring that our methods are reliable and consistent. Additionally, our process capability is now at or above 1.33, indicating a highly capable process that produces quality results within specifications. When it comes to dry syrup sampling, we've optimized our approach to make it more efficient. Traditionally, sampling involved taking 20 samples of 5 grams each, totaling 100 grams. While effective, this method was quite resource-intensive. Our new optimized method reduces the number of samples to just 8, still at 5 grams each, which means only 40 grams are needed overall. This not only saves material, but also speeds up the process without compromising accuracy. Overall, these improvements reflect our ongoing commitment to process excellence and efficiency, aligning perfectly with our broader goal of continuous improvement in quality and resource management. Now, let's take a closer look at some control strategies we use to prevent excess sampling. These are key approaches that help us strike the right balance between thorough process monitoring and efficiency. First up is Process Analytical Technology, or PAT. PAT allows us to analyze our processes in real time, providing immediate insights without waiting for traditional lab results. Then we have Statistical Process Control, or SPC, which uses statistical methods to monitor process stability and detect any variations early on. Next, there's Historical Batch Trend Analysis. By reviewing past batch data, we can identify patterns and anticipate issues before they happen, making our process more proactive. Continuous Process Verification, or CPV, is another essential strategy. It involves ongoing validation of our process performance, ensuring everything remains within specified limits throughout production. Lastly, automated sampling systems play a vital role. They reduce the need for manual sampling, which not only saves time, but also minimizes human error. All of these strategies work together to reduce the frequency of manual sampling, but at the same time, they keep our process under close watch. This integration is critical to maintaining high quality while improving efficiency across our operations. When implementing reduced sampling, there are several key documents we need to have in place to ensure regulatory compliance. First, a comprehensive risk assessment report is essential because it helps us justify why reduced sampling is appropriate for our specific process. Next, the sampling SOP, or Standard Operating Procedure, provides detailed instructions on how sampling should be conducted under these new parameters. We also need a process validation report, which demonstrates that our process consistently produces quality products even with fewer samples. Statistical justification is another critical element. It involves providing the scientific rationale and data to support that reduced sampling still maintains product safety and quality standards. Change control documentation is necessary to record and approve any modifications to sampling procedures, ensuring traceability and compliance. Lastly, an annual product quality review helps us monitor ongoing performance and verify that our reduced sampling approach continues to meet regulatory expectations. It's important to remember that inspectors will review the scientific rationale behind our sampling frequency. They want to see that our decision to reduce sampling isn't arbitrary, but backed by solid data and thorough analysis. This review process is part of how regulators ensure that our product quality remains uncompromised, even as we optimize our sampling strategies to be more efficient. Let's take a look at the progress we've made in reducing batch wastage. As you can see, the total batch wastage has decreased from 0.26% down to just 0.095%. That's a significant improvement. In fact, we're looking at approximately a 63% reduction in wastage. This isn't just a number, it's a reflection of our team's effort to optimize processes and minimize waste. When we consider the overall reduction in sampling wastage, it aligns perfectly with our goal to improve efficiency and sustainability across the board. These numbers highlight how targeted improvements can lead to substantial benefits, not only in cost savings, but also in our environmental impact. This slide really underscores the importance of continuous monitoring and process refinement in achieving our operational excellence. In conclusion, adopting risk-based and statistical optimization for IPQC sampling offers several significant benefits. First, it allows us to drastically reduce sample wastage, which not only conserves resources, but also streamlines our process. This approach also helps lower manufacturing costs by making our sampling more efficient. Additionally, it enables faster production monitoring, so we can identify and address issues more quickly. Importantly, this method ensures we stay compliant with global GMP guidelines, maintaining high standards across our operations. Now, to achieve these outcomes, there are key success factors we need to focus on. Robust process validation is essential, making sure our processes are well understood and reliable. Statistical justification is also critical. We must base our sampling decisions on solid data and sound analysis. Continuous monitoring helps us stay proactive, catching potential issues before they escalate. And finally, maintaining a strong quality risk management system underpins everything, ensuring that quality is integrated into every step of our production. Together, these elements create a comprehensive framework that aligns with our overall goal, delivering high-quality products efficiently and consistently while adhering to the highest standards. For further support, please don't hesitate to contact us. We're here to help with any questions or additional information you might need. Remember, our goal is to ensure you feel confident and well-informed about everything we've covered today. Thank you so much for your time and attention. Your engagement truly makes a difference. And again, if you need anything else, just reach out. Thank you.